Genetic technologies are tools and techniques that use knowledge of DNA and inheritance. Some genetic technologies are used to read, analyze, or compare genetic information. Other genetic technologies are used to change genetic information in cells or organisms. These technologies are used in areas such as medicine, agriculture, conservation, and forensic science. One important genetic technology is DNA sequencing. DNA sequencing is used to find the order of bases in a DNA molecule. This can help scientists identify genetic differences, study inherited conditions, and compare genetic information between organisms. Genetic testing is another important technology. Genetic testing can be used to look for changes in DNA that may be linked to inherited conditions, ancestry, or other biological information. DNA profiling is a genetic technology used to compare DNA samples. It is often used in forensic science. For example, scientists may compare DNA from a crime scene with DNA from a known person. DNA profiling can also be used in missing persons investigations or to help identify biological relationships. Genetic engineering involves altering the DNA of an organism to produce a desired trait. For example, scientists may use genetic engineering to give a crop plant resistance to pests or disease. Gene therapy is used in medicine to treat, prevent, or sometimes cure disease by adding, replacing, or changing genes. CRISPR is an example of a gene editing technology. It can be used to make specific changes to DNA. Some genetic technologies are already current, meaning they are used regularly in science, medicine, agriculture, or forensic work. Other technologies are emerging, meaning they are still being developed, tested, or used in more limited ways. As these technologies become more powerful, people need to consider both their benefits and possible risks.